Hello everyone, welcome to this webinar. Unfortunately, there is an issue with the platform with GoToWebinar, so we are not able to activate our cameras. So today you will hear us, but you cannot see our faces. So we are not happy with that, but this is what it is, and we hope that you will enjoy in any case this webinar. So before starting the presentation, you will be asked to ask questions. Please click on the icon in the toolbar in order to address your question in the final part in the Q&A session. A recording of today's webinar will be available after the session for your reference. And a brief feedback survey will appear at the end, so please take just 1 minute to complete it. Your input is extremely valuable and helps us improve future sessions. So let me introduce the speakers. I'm Paola Vigilanza. I'm Global Solution Manager for Pyrogen Testing for Biomerieux. And here the presenters today will be Alessandro Pauletto. Alessandro Pauletto is graduated in life science. He started his activity in the world of endotoxin in 2005, covering different roles. For almost 20 years, he has been supporting many companies globally in addressing regulatory and technical issues in the world of endotoxin and pyrogen detection. And he currently holds the position of scientific and regulatory subject matter expert in pyrogen testing. The second speaker today will be Dr. Christian Faderl, project leader Endotoxin Services. He holds a PhD in organic chemistry from the University of Regensburg. After experiences in the quality management in pharma, he currently serves as project leader working in the R&D department for endotoxin and bioMerieux. He is responsible for all endotoxin service projects and is recognized expert for low endotoxin recovery and endotoxin demasking problems. Together, they will provide both scientific insights and real-world experience on the mechanism behind LER, the regulatory implications, and the practical strategies for mitigation of LER. So thank you for joining the webinar. During the webinar, Alessandro and Christian will try to address the problems regarding LER, and please, if you find, yes, before we begin, we have just, before starting the presentation with Alessandro, we kindly ask you to respond to this quick poll, which is about your experience with the LER phenomenon. If you already addressed it internally or externally, if you will need to take action soon, or if the topic is not relevant for your production. Thanks for participating into the poll. Take some seconds to respond, and then we are ready to start with the part 1. Just a few seconds. Okay. I don't know if you can see on the screen the results. So Many of you are required to conduct such studies and need or would like information to learn more about LER. Many of you perform in-house LER studies. Some of you outsource LER studies, 13%. For 13% of you, the topic is not relevant. So I leave the land over to Alessandro with the first part of the presentation regarding the regulatory approach and the biochemistry behind LER. Please, Alessandro.
Thank you, Paola. You have to assign me the control. Okay, thank you, Paola, for your kind introduction. And good to see that we have many people involved or interested in long-dose recovery. And I think we will cover a lot in this webinar. Oh, okay. Let me start from a regulatory point of view because, as I'm used to say, most of the time in a regulation we can find a reason why we do something in our labs. So this is a guideline. This is a question that you can find. Sorry, could you come back? What is going on? Paola, could you come back to the first?
I'm not managing it, so
Me neither. So I don't understand why you move
don't
on.
know why, honestly. Let's go back. Sorry for the audience. Okay.
Could you come back to the first? Okay, that's great. Thank you. So as I said before, this is a question, the question number 3 that you can find in the FDA guidance since 2012. Okay, and as you can see here, for the first time, we see something related to the impact that you could have in terms of endotoxin detection by storage and handling your sample. So this is a very important and relevant regulation behind the loan dose recovery, and then we will see better after this this slide. Also because in the next one. Okay, we see that there is a similar wording, but just 3 years later on the previous one, only 3 years after the FDA guidance related to BT, we have a similar definition in terms of impact of the storage in toxin detection. We can see that in European Pharmacopoeia in the chapter 5.1.10. Okay, so this is very important because for the first time we see that in European Pharmacopoeia, and okay, and I said all I presented you a couple of slides about what could be the impact in terms of endotoxination by storage because maybe strange, but only 1 year after the 2012 FDA guidance related to bacterial endotoxin test related to the impact of the storage in endotoxin detection, we had the first speech reporting low endotoxin recovery. The first speech was brought by Chen Winter to the PDA Congress in the US, and they have seen for the first time the issue related the low endotoxin recovery issue. Okay. Oh, at the time, the definition of a low tox recovery was a time-dependent phenomena in which the true content of toxin in biological product with a specific formulation could have an issue related to underestimation, okay, by bacterial endotoxin test, whatever it is. At that time, there was a specific theory behind. The theory behind was the sample formulation could create a sort of long dose recovery complex with LPS, and that complex could not allow to be detected by factor C. So by or through this complex, LPS could be not detected anymore by the factor C. Okay. Um, the first official definition of what is low interest recovery was in 2019. We see for the first time in 2019 in the PDA Technical Report 82. At the time, the definition that is still available or whether it's still applicable is an ability to recover at least 50% in activity of over time. This is a critical point when no amount of endotoxin is added to an undiluted product, and we will see later on by Christian the meaning of that. PDA Technical Report 82 is still the main reference, also in terms of a regulatory point of view, because inside this specific document you can find many different information related to a definition of what is LER, how to address and how to fix if you have any kind of low interest recovery issue. So I said before the official definition of what is a low interest recovery, but maybe it's better to provide you an example. Think about to have a specific product and think about to make a spike to add a specific amount of endotoxin in your drug product. And then you start making the sample preparation and testing your product. Think about to do that at different time points. Usually, if you don't have any kind of low interest recovery issue in your sample, you should see that the endotoxin content in your sample should stay stable. So you should see a little variation, but always within the certain level or certain range in LER or recombinant factor C detection. If you add a low interest recovery issue, you should see something like on the right side of this presentation. You will see that there is a little decrease of the signal till reaching a value that is below 50%. In this specific case, we can define that you have low interest recovery. So when you change, when you have change of the toxin amount in the sample over the time, in this case, you have what is defined as low interest recovery. Very important point is many times when I discuss with my customers, I see there is a lot of misunderstanding. There is a strong misunderstanding between what is a low endotoxin recovery and what is low spike recovery. Even if the wording looks like to be very similar, they are different. They are completely different. Low spike recovery, as you know, the spike recovery is a way in a kinetic method or in quantitative bacterial endotoxin test to define the emission or enhancement that you can have in the reaction. So it's a way to check the interference level that you have in your sample reaction. In low spike recovery, when you test your sample and you have a low spike recovery, what you get is a spike recovery that is below 50%. But as you know, low spike recovery below 50% give you a invalid test. So when you have a low spike recovery, you have an invalid test. An invalid test is a test that is no test, means that you don't have any kind of product release. So there is no issue in terms of quality. It's just an invalid test. When you have a low interest recovery issue, you have a completely different issue because all the situation is related to the endotoxin amount you are able to detect in your sample. In this case, the main problem that you could have is a valid test because the spike recovery will be always within the range of 50 to 100%, so always valid. So the test is still valid, but you could release something that is a sort of false positive result. So because you are releasing something where you are not able to detect anymore the real endotoxin amount you have in your sample. This is completely different to what is low spike recovery. As I said before, invalid test against false negative results. This is a strong difference. What is the current main theory behind the mechanism in low interest recovery? As you know, LAPS is LPS structure is based on a monomeric form. The monomeric form has a weight that usually is between 10 to 20 kilodalton. But in nature, in a real situation, your sample, is not possible to have a monomeric form of LPS. Usually you have a big aggregation state. So you could have many different LPS, all of them in a single aggregation state. So usually you have something like that is between 500 and 1000 kilodalton. Okay. Um, when you have in your sample a chelator and a surfactant, you could have the low toxic recovery issue. Why? Because by chelating agent, you could remove the divalent cations that has a specific function in LPS structure. The chelator or the chelating agent could remove the divalent cations from the structure. In this way, you could have a little decrease of the size because the single monomeric form could be removed from the aggregation state, the bigger aggregation state, and then the surfactant complete the action. So in this way, you could see a reduction of the structure of the LPS. So this is why you have a long-term recovery issue based on the chelating agent plus a surfactant. Another misunderstanding behind low-interest recovery is the difference between low-interest recovery and LPS adhesion on surfaces. Low-interest recovery, as we have seen before, is a phenomenon related to the matrix itself. So it's the matrix that has an impact in terms of decreasing the size of the LPS structure. Decreasing the size make endotoxin to have a lower activity. Okay, so this is clearly due to the matrix of the sample. The LPS adhesion is something completely different because it's related to the sample container and should be addressed a completely different way. So all-time studies should not take in account this issue because you should have already fixed this issue because there are different kinds of regulation like European Pharmacopoeia Chapter 5110, where it's clearly written that you cannot use any kind of accessories where you are not sure to have to have issue related to interference and adhesion of LPS to the surface. At the same time, also on USP, currently there isn't, but there was a specific sentence about this issue in the draft 2024 draft of the new 1085. Not sure that this sentence or this statement would be part or could be part in the final version, but in the draft it was, and was clearly written that you should perform some long-term study without any specific matrix or on water in order to be sure that the sample container has no impact in terms of the toxin detection. Okay, so this is a completely different phenomena. It's not LER because due to the sample container, not to the matrix. LER is always related to matrix, sample matrix. And last but not least, another misunderstanding was in all-time study. As you know, there are two different kinds of all-time study. The first one is the QC all-time study that is related to the time between sampling and testing. Most of the time, the QC all-time study should be done at a temperature that could be between 2 and 8 Celsius degree because most of the time when you take a sample, when you do a sampling, you keep the sample in the fridge, you store the sample in the fridge clearly. So this is why a QC time study is done at this specific temperature, and most of the time between sampling and testing is very, very low. Is I would say not more than 2 or 3 days. Indeed, a QC time study is usually takes not more than 3 days usually because you have to take in account temperature and time relevant for this specific phase of your process. In a low and toxic recovery study, all-time study, as written in pediatric report 2, and this will be discussed more in detail by Christian. In this low-dose recovery all-time study, you should take into account the manufacturing process. So this means that temperature and timing, if you compare to QC all-time study, will be completely different because it is very difficult to find a manufacturing process that is done or is performed at 2 degrees. Usually, this is done at room temperature, and usually takes more than 2 or 3 days. Usually takes, I would say, 6, 7, 8 days. Okay, so this is why a long dose recovery all-time study usually has a completely different timing and temperature if you compare to all-time study. And what is mandatory today is only long-dose recovery all-time study, speaking for long-dose recovery. A QC all-time study remain always something that is required by GMP usually. So let's discuss why we have to perform long-dose recovery all-time study. We have to perform a long-dose recovery all-time study because it's specifically required by FDA when you have a biological product. I don't know why. Paola, could you come back
Yes.
to the go back? Another
Okay.
one.
Slide 17.
Another
This
one.
one.
Another one. The previous one. Okay, perfect. Okay, so long-term recovery all-time study is required in BLA, okay, because a specific requirement by FDA. Even if this is for FDA, we can think about what is required in Europe. In Europe, there is something similar to what what are required by FDA. Also in Europe, we have something where it's written that we should perform low-dose recovery all-time study if we have a biological product where we have a specific formulation, usually by a combination of surfactant and chelator. Okay, and in this specific case, in order to submit marketing authorization, you should add in this marketing authorization also a low-dose recovery all-time study. Okay, this is written in the EMA website related to the question answer for this specific topic. Very important point here is clearly written that you should consider as main reference for performing all-time study and also for checking if you have an issue in terms of long-term recovery, what is written in pediatric report 82. So even if it's not a real a document from a public authority, is still something that must be followed because there are some specific reference to this specific document, as I said before, written by PDA. And this is my last slide of my presentation, and I let to my colleague take over the presentation. Thank you, Christian.
Thanks, Alessandro.
Thank you very much. I hope you can hear me. Unfortunately, you can also not see me, but just look at the picture at the beginning of the presentation and you know who I am. Hello everybody. I try now to, yes, it's working, great. So I will present to you today how to perform an LER study. I saw in the first question we sent you that a lot of people, a lot of you are doing LER studies already. So therefore, you might know how to do this and so on. So you can relax at this first point, so to say. Then I will go a little bit into more detail in endotoxin demasking, a general approach, and some case studies I will present. And then I will switch to the EndoRS kit, which is one special sample preparation kit for endotoxin demasking, and finally some case studies from industry on that part. So how to do an LER whole time study? Why are you doing it? So Alessandro already presented why you need to do it. For me personally, it's 2 points. The first one, patient safety, that you really can detect if there is an endotoxin contamination. And the second one, of course, and very often, very often the more important part, to get the go from the authorities that you can use your endotoxin test and that there are no issues and you can go on and release your product. In the technical report 82 is a guidance, are some points described which are important for such an LER whole time study. And the authorities usually are looking also after this and are accepting this when you're doing a whole time study or an LER study according to these guidelines written in the technical report 82. Nevertheless, I included some of my comments to that and from our experiences, some things which might be maybe important or to have a look at this. So the first point, LER studies should be done with a final drug product, of course, but still, whenever you're doing an endotoxin test, you want to make sure that you can detect an endotoxin contamination. So therefore, it does not really start and end with the final drug product. Therefore, whenever you have a test implemented, you want to know if this test is working. Otherwise, no endotoxin test needed if it's always negative, so to say. But of course, for authorities, for the FDA, for instance, final drug product is the most important part. And then the second one, the second point here is with which endotoxin to do the LER study. RSE, so the reference standard endotoxin, or controlled standard endotoxins are accepted most of the times from the authorities, and naturally occurring endotoxins can be used, but not only. So therefore, stick to the RSE and the CSE. From our experiences, we know that sometimes the FDA or other authorities can challenge you on why you use this controlled standard endotoxin or that controlled standard endotoxin for your LER study. And that's why we performed a small study on this. So we did some LER studies with different CSE and also RSE, of course, and saw also some differences and different behaviors of different CSE. Therefore, I put it here in the reference. So we presented this study at PharmaLab last year. So I'm sorry that it's wrong here. It's written 2025. That's not true. It was 2024 at the PharmaLab, so last year, where we presented this study. And therefore, our recommendation is use the RSE for the LER studies, then no questions asked from the FDA, so to say. Then there in this technical report, there is no real rule or no real guideline which endotoxin, so which spike level you should use. So it's written that you should use a spike level that after sample preparations or after the elution of the product, you are within the standard curve range, in the middle of the standard curve range. But actually, many authorities are looking, of course, to the patient safety and are also requesting to do this LER study with the endotoxin limit. So not all of them, of course, but we heard this from time to time that the endotoxin limit is quite important here and to spike the product where the test is, so to say, important, because we also know it could be that when you spike a product with, for instance, 100 EU per milliliter, after 7 days at room temperature, you still detect 60 EU per milliliter and you have no low endotoxin recovery effect. But actually, when you spike with your endotoxin limit of 5 EU per milliliter, for instance, this is gone immediately and then you have an issue. And to prevent such discrepancies, I would recommend to use always the endotoxin limit for your LER study. Then, of course, you should not dilute the product with a spike, so with the endotoxin spike. And you should use, Alessandro said this before, you should use appropriate containers for your LER study, that the containers are not interacting with the endotoxin you add. And always use a water control in your LER study, because finally you need a reference to calculate the recoveries to something. So therefore, oh, what happened now? Did I do that?
I don't know. Let's just one moment. Yes. Is okay?
Yes, it's working again. Thank you. Yes, so therefore use a water control where you can refill the endotoxin, so where you can then calculate the endotoxin recoveries too. Last but not least, for a for the biological license application, for instance, you need to test or to do this LER study on 3 different batches. Then the second point, the second part of this LER study is storage. And here the LER study should be done at process relevant temperatures and times, as Alessandro already pointed out. So when you have a process step at room temperature, you should use the worst case scenario and do the LER study at room temperature. Also regarding the times, when your production time takes 3 days and 2 additional days until it can be tested, also with the risk assessment and so on, then take this this worst case scenario for your LER study. Also mentioned in the technical report 82, if there are some inconclusive results or some discrepancies, you may need to extend your LER study, maybe for 1 additional time point if there was everything above 50%, but the last point tested was below 50%. You need to extend your LER study and test at least 1 more time point. Or maybe if there is an LER issue, you solve the LER, then we also saw once that the authorities requested to include also the sample hold time into this LER study to have really mimicked the whole production process and test process, that in the whole that it can be that the sample still is in the QC lab for 2, 3, 4 additional days, and this was then also requested by the FDA to add this to the LER study because there were some issues before. Last but not least, the test, and there, yes, as I mentioned before, you need to calculate your recoveries to something. When you use the reference endotoxin for spiking, of course, there's always the possibility to use the theoretical value which you used for spiking, but I would highly recommend to use, and it's also written in the technical report 82, to use the water control. And here there is no real rule if to use the water control at time point 0, so at the time point when the LER study started, or to use the water control at the measured time point of the sample, so the time point X water control, to calculate the recovery. So that is not so clear. So you need to be aware of this prior to start the LER study. And at least you need to cover 4 time points in your LER study to have valid and accurate results. There are 2 possible modes of how to do an LER study. So here is shown one of these modes. It's the online mode, where you spike endotoxin to your drug product and also to the water control, of course. You aliquot the product and the water control, and then you measure at every time point. So in this example, you measure then 4 times and get the results, where you can then draw a nice LER, so a nice LER graph, so to say. Here, the benefit, of course, you just need to spike once, but the drawback, you need to measure several times. And there is then the second possibility to do an LER study in a reverse mode or a reverse manner, where you spike first the time point 0, then you put the sample for storage, then you spike 1 day or 2 days later the time point 2, put it to storage, and then time point 1, and finally at the time point 0 where you spike then the water control and maybe also the drug product. You measure every sample all together. Benefit, of course, you just measure once. Drawback, you have to spike more often the drug product and the water. So there are no guidance, so to say, which version you use. Then, here, now it's working. LER. So here are some examples how such LER curves can look like. So these are no real data. This is just for visualization on some curves how an LER study could look like. So LER curve 1, you can see that you have a steady lowering of the endotoxin values you recover, and so this is a typical LER curve, LER kinetic for maybe such citrate and tweens or chelator and surfactant buffers. The same for the LER curve 3, where you have an immediate drop after time. So when you measured at time point 1 directly, no recoveries at all anymore. So these are more typical LER issues. Then for LER curve 2, it's not a really typical LER kinetic. There is something else might be the reason for such curves or for such studies. I will go into detail in the next 2 slides into this one. And then there is maybe a slight LER, which you can see for LER curve 4, where you start for 100%, but then over time you are slightly below 50%, unfortunately, but this is an LER issue which you need to address. And now what you were waiting for, general approaches for endotoxin demasking. So you detected an LER, what's next? I would recommend first to do a root cause analysis, because you need to know where the LER issue is coming from to do a proper demasking development study, so to say. Then second is changing the sample preparation to reverse the LER effect. And the third point would be then to repeat the LER study with a new sample preparation. Sounds easy. Sometimes it is as easy as it sounds, sometimes not. So first of all, root cause analysis. Why? Because it can be important for the further studies and experiments to know what the reason for the LER is. Do I also have to look at the drug substance? Do I have to look at some in-process controls? Maybe I have a different issue of interference and it's not really low in toxin recovery, but it just looks like that. Maybe it's the container which gives you the error. Maybe it's how you did the sample preparation or how you set up the whole LER study. And there I come back now to this example of the LER curve 2, which is a little bit uncommon for an LER, a typical low in toxin recovery effect triggered by the media, by the buffer, by the drug product, because you have an immediate drop of endotoxin and then it stays around about at this level for the whole LER study. So here it could be that that there was an error in the sample preparation. Maybe a strong dilution was done. Maybe the container had an effect, as Alessandro said before, that their endotoxin bound to the container. Maybe there was also something wrong with endotoxin spike, or the control scented endotoxin or something did something strange here. So this is not a really typical LER issue. And when you know where it's coming from, the LER effect, you know how to handle this. Second point, so you have now detected low in toxin recovery. What can I do next? And there are also some guidelines and some advices in the technical report 82, where so there are some things written what you could do, also some case studies on that. And I also highlighted some things which worked from time to time, also in our hands, which are important to test, of course, and which you should test. for instance, to change the sample pH, especially when the pH is out of specification here or out of range, then to add some divalent cations to the sample for sample preparation, for instance, magnesium sulfate or something like that, because it's assumed that divalent cations, so in the mechanism also Alessandro showed, it's assumed that the divalent cations which stabilize the LPS, the detectable LPS structure, are not there and also not there anymore, cannot stabilize the endotoxin structure, and therefore by adding additional divalent cations, maybe the detectable structure of the endotoxin can be reformed. Also, it's worth to try some dispersing agents like pyrospores to get rid of interferences of LER. Also, some surface active components, maybe to block some parts of the molecule which could interact and bind to endotoxin. Also, sometimes helpful to use organic solvents for sample preparation. And also literature known in some cases, if the enzymes bind to the LPS, to the endotoxin, it can maybe help to digest them, to make a proteolysis of this, and therefore then release the bound endotoxin. Very often, if you have such an LER curve 4, as it's shown here, when the LER issue is not so big, very often or usually such sample preparations, such easy and fast sample preparations, I call them, is very often helping to reverse this LER effect. And here I have a case study for you, so a real study we did in our labs for an antibody. Here we had a very low spike of 0.5 U per milliliter. We spiked with a control standard endotoxin and had a hold time of 7 and 11 days at room temperature. The current sample preparation was 1 to 20 dilution in water endotoxin free, and then detected with the endozyme 2 assay. So that is an RFC assay. And we had a slight LER effect, recoveries of 44% and 38%. And here then with a simple change of the buffer, we could reverse this LER effect. So by changing the buffer or the solvent from water to phosphate buffered saline, 10 millimolar, we could recover endotoxin to after 7 days 85%, and after 11 days 106% recovery. So very nice. By just exchanging this solvent, the LER issue was gone or reversed, so to say. Now the second, yes, here it is. The second case study I want to show you is a similar one, also an antibody with 5 U per milliliter reference standard endotoxin spiked, also detected with the endozyme 2 assay, and the current sample preparation was a 1 to 40 dilution in water endotoxin free, and after more than 7 days at room temperature, 39% recovery, so a slight LER effect again. And after some experiments, we could reverse this LER effect by changing the solvent, and in this case also the dilution factor. We could recover the 5 U per milliliter RSE quite good. So with a sample preparation of 1 to 150 in 5 millimolar PBS, we could get more than 50% back at the worst case scenario, 7 days at room temperature. So that are our studies where we were successful with the PBS buffer. So the background for that is maybe because LPS is very often decorated with phosphate groups. If they are part of the whole LER effect of the endotoxin masking, by introducing more phosphate to the buffer, it could be that we saturate the product and the active components which interact with the phosphate, and therefore release the endotoxin and make them detectable again. And here I would have one question for you. If you were also successful with LER, so to reverse the LER effect by just changing the solvent to a normal, so easy buffer, so to say, like magnesium sulfate and so on and so on. If this works every time or not, that would be great if you would comment here.
Yes, thank you, Christian. Attendees are responding. Just wait 30 seconds more before terminating and sharing the results. Okay, you see the results on the screen?
Yes, it is 50% roundabout had never observed an LER issue. Then also 50% roundabout, so not completely 50%, yes, but not in every case. And 2 persons said yes always, and 2 persons that did never work. Thank you very much. So that is also our experience, that very often this helps and this works, but sometimes the LER effect is too big here. Paola, you might need to give me the presenter again.
I think, oh, I don't know why it removed your control. Now you have the control again.
Thank you very much. Yes, and that leads me to these 50% which said, okay, yes, but not always. That leads me to the NRS kit. Because if this is not working, so if all the things which are easy, so to say, so it's also not easy, of course. It's a lot of work, a lot of experiments which needs to be done to test all the different possibilities and all the different buffers which are there. But if all of this was done and it doesn't work, it doesn't help, then there is still the NRS kit, and this is what we would do then as well from our point of view. If the easy sample preparations are not working, then we go to the NRS kit. And the NRS kit is a toolbox. It's a little bit more difficult and complicated to work with that than to just dilute the product in certain buffers, because it's not a ready-to-use toolbox, so to say. So not a ready-to-use buffer. So you have in this NRS kit 6 different buffers who can be useful or not so useful to demask endotoxin in your product. So all of these buffers should, or some of these buffers should interact with the endotoxin, should release the endotoxin from its masked state, should stabilize the endotoxin and bring it back to a detectable structure that the endotoxin can be detected in your product. And all of this, of course, in the sample, so during sample preparation. Here I have also case studies for you on typical LER buffers. On the left, you see we did an LER study with a 10 EU per milliliter RSE, whole time study at room temperature with 10 millimolar citrate and different concentrations of polysorbate 80. So these are really well known or is a well-known LER buffer. And on the right side, you see the LER study, also 10 U per milliliter RSE masked in 1 time, so 10 millimolar PBS plus different concentrations of polysorbate 80. So there is an LER effect, of course, with the citrate and polysorbate stronger than with the phosphate polysorbate, but in both kinetics, so you have LER or you are below 50% after 7 days. And with the NRS kit, so with the sample preparation I show you here, by addition of component B a pre-diluted component E, so 1 to 5 pre-diluted. Then an incubation time follows to bring the equilibrium, so to give it the equilibrium, to lead to an equilibrium. Sorry for that. Then a 1 to 10 dilution in water, followed by the endolyzer assay for detection. So the endolyzer assay is an RFC assay that led to a good recovery, around about 100%. So we could demask the endotoxin in these buffers quite good with just 2 of the NDRS components, B and E, 1 to 5. In the second buffer, you see that we needed completely different sample preparation to overcome the LER effect. So even when the masking mechanism is quite similar, so here also it's like the PBS catches the magnesium ions, and then the polysorbate masks the endotoxins, also builds vesicles with endotoxin. In this case, here we need a completely different sample preparation. So we need to add component A, component C, and the pre-diluted component E, 1 to 20. Again, 1 to 10 dilution in water, and then detection with the analyzer assay. So similar masking mechanism, but completely different sample preparation with the NDRS kit. And that is the takeaway I want to give you here. So you need to develop for your own product with your own masking condition. You need to develop your own separate sample preparation. And even here with the same buffers, you see the lower the polysorbate 80 concentration was, the worst was the recovery. So here just 60% recovery. So maybe even here for this last buffer, we might need to change the sample preparation a little bit. Usually it's the component E which might need to be changed here to get better recoveries. But as you see, even when the buffer is slightly changed, so the formulation, the product slightly changed, it can have a complete, so a big influence on the recoveries and on the protocol you need to use. So yes, yeah, and of course the NDRS kit comes with a package insert, and in this package insert, the workflow is described quite good. But just for your information, or what I want to highlight is that the endotoxin demasking consists not only of the NDRS components you add, so that is the most important part, of course, where you add all the NDRS components which are necessary, but also the incubation times can be important. Also, the dilution can be critical, the dilution step after addition of the NDRS components, and also the endotoxin detection can be critical and crucial for endotoxin demasking. And I want to not stay too long on this, but just highlight some things, some critical things which can be or should be developed during such a sample preparation or such a development project with the NDRS kit. For instance, of course, first of all, the NDRS components. Some are beneficial, some are not so good for endotoxin demasking in your product. Then, so there, this is something you need to screen. Second, the dilution step. Sometimes the dilution can reverse the demasking again and lead to additional masking, or with the dilution you introduce, which you need to do to get valid results, so sometimes one solvent is better than the other solvent, so to say. So this needs to be tested and checked. Also the incubation times, 30 minutes incubation time after NDRS component addition, or also after the dilution step, do you need an additional incubation time? Yes or no, something like that can have an effect or can be critical for endotoxin demasking with the NDRS kit. And last but not least, the energy you bring into the demasking shaking steps. We have a lot of shaking steps during NDRS demasking. Sometimes this can be critical. Doesn't need to, but can be critical. And last but not least, the detection assay. Originally, we developed the NRS kit in combination with the endolyzer assay. So that is a very special assay where the plate is coated with a endotoxin binding phage protein. And we know that with this separation, so with this additional sample preparation of the endolyzer kit, we get very often faster and better results than when we combine the NRS kit with conventional RFC or LEL assays. So that is also something to keep in mind, that also the detection assay can play a critical role in endotoxin demasking with the NRS kit. And here just one example how it can, so how different the results can be. So when you do an endotoxin demasking and combine the same samples with a chromogenic LEL assay or with the endolyzer assay, you see on the bars, so on the columns, that with the endolyzer assay we can detect quite good. So in green, endotoxin, which we recovered with the NRS combinations, and on the left there are no bars coming up because the demasking did not work with these with this LEL assay used. So the change of the detection assay can play a big role for endotoxin demasking, especially with the NRS kit. And I see the time is running, so therefore I'm showing the case studies a little bit faster. So there is one case study described in the PDA Technical Report 82. It's the case study 7, where a successful endotoxin demasking with the NRS kit is described in combination with the endolyzer assay, but also with the LUL assay. So first, the demasking was done for this sample in combination with the endolyzer assay, but then when the demasking protocol was developed, it was then adapted to the LUL assay successfully. And as a second case study in industry, there was a presentation at PharmaLab 2022. There were actually 2 presentations where first the whole LER challenges and the solutions were shown, and then in the second presentation, the validation of this solution was demonstrated of a monoclonal antibody, 6 EU per milliliter, and with the NRS kit in combination with endolyzer assay, the LER issue was reversed, and the FDA approved this method. And then last but not least, so whenever you decide to do endotoxin demasking, even if you're not interested in the NRS kit, because this is really what comes after the easy sample preparation, so the fast sample preparation, so to say, bioMérieux can support you or wants to support you, of course. So we have endotoxin service, first feasibility studies where we show or demonstrate if the endotoxin detection assays from bioMérieux are capable to detect or can be used for your products. And then we have all parts of services for LER and endotoxin demasking. We do whole time studies, LER studies, but also test for demasking with the NRS kit and do whole demasking development projects with the NRS kit if NRS is really needed for your product. And always, when you want to use NRS as well, please reach out to us, and we can discuss with you and support you with that. Thank you.
Thank you, Christian. Thanks, everyone. So take just some seconds to respond to these, and we will reply to. We received many questions. Already some have been replied in the chat, and we will take the opportunity to respond to some of the other questions. Please, before disconnecting, stay connected because you will receive a poll regarding the feedback that you have for this webinar you attended. So I can read Alessandro, Christian, feel free to reply depending on your thoughts. First question for you: were companies able to successfully defend their positions who were questioned on the use of CSE instead of RSE?
Christian, if you want, I can take it wrong.
Yes, of course.
Okay, according to PDA Technical Report 82, you could use both RSC or CSC, so should be acceptable to use all of them. What is important is you should use, let me say, uh, we have shown in a particular speech that different kind of CSC could have a different kind of results. So what is important is you should have clear in mind that the CSC you are using is not designed to be used for all-time study, because it's not the worst case as it is LSC. So can be used, but not because different to LSC. Okay, so this is my reply to that. I don't know if you want to add, Christian, something.
No, that's good. Thanks.
Second question for you, which is what is the minimum all time required in an LER study?
Maybe I take that. It depends on your manufacturing process. So I would say from even from in-process control to in-process control, it can be somehow defended in front of authorities, of course, that you take this as an LER study time. But of course, you should also keep in mind that not only the sampling is then the time point of the LER study, but the actual testing itself. So always be aware of when can this sample then actually be tested in the QC lab, and to also take this into your calculations, especially for authorities, to really show them directly, okay, here from this in-process control at this time we measured it until here, until we measured it. But to be on the safe side, I would say 7 days room temperature is always a very good time point to take.
Okay, quick question. This is to be addressed very quickly. Does a lower endotoxin recovery study need to be conducted in the original primary packaging, or can it be done in another container?
I can say my opinion on that. Again, according to PDA Technical Report 32, the low endotoxin recovery all-time study should be done in a container that is suitable for such as study. Okay, so it means that can be the primary packaging or can be any other kind of container that is suitable for low endotoxin recovery all-time study. As I said before, as not to be as not to have any kind of issue in terms of interference. But we advise also to check, even if you don't have anything in terms of all-time study in a specific container, I would like to try also in the primary packaging, just to have the full reply. But anyway, from a regulatory point of view, it's not required clearly to perform in the primary packaging. So you could use whatever you think is acceptable in terms of low toxicology all-time study. But the sample container must be suitable for this kind of use.
Okay, thank you. Just two additional questions, last two. First one, what is the experience next steps if also the Endo-RS kit cannot overcome LER? This is a tricky question.
Yes, of
Christian?
course. So that depends, of course, on your policy, so to say. So I would say if also the Endo-RS kit is not working and so on, you could have the possibility to go with the MAT, so with the MAT, to test on pyrogens, but that you then of course need to do for release test all the time, so to say. So not only 3 times and that's it. And it could be that the MAT also shows LER. So that is something we also saw, that for some products the MAT could be an alternative, but for the most of them, if there is LER in the BET, it very likely also shows LER in the MAT, and that is then not always a solution.
Thank you.
Yeah, sorry.
Sorry. No, the very last question, because we are out of time. We have a contact, not sure to clearly understand the difference between LER holding time and QC testing holding time. Which data need to be generated to cover the QC testing holding time?
Okay, can try to repeat what I said before. QC ultimate study should take account only the time and the temperature related to the time between when you make the sampling to the time to when you perform the testing. Okay, so you have to take account only this limited amount of time, usually just a couple of days. Okay, because usually maybe you can take the sample on Friday and you test it on Monday, for example, and usually it's done as usual. The difference is in time and temperature, and there are other differences, but mainly is in the time and temperature, because in a low dose recovery ultimate study, you should take in account really the temperature and the time related to your manufacturing process. That is completely different. So you have to provide data always related to different time points in terms of detection level, but time and temperature will be completely different, because usually manufacturing process is at room temperature, usually takes more times, and always you have to consider to test your drug product for sure. But what is different is time and temperature, I would say, and other details that we don't have time to discuss here. But this is the main difference.
Okay, I recommend eventually to keep in touch with you, Alessandro,
Yeah, sure.
to get more clarifications if any doubts are there still. So since we are out of time, we received additional questions, but we will answer by email. So thank you for participating into this webinar. Take one moment to respond to the final survey regarding the quality of the webinar, because it will help us to improve our and your experience as well. Thanks, everyone. Thanks, Alessandro. Thanks, Christian.
Thank you.